Okay, so this part is about how you can access our data. How can you retrieve our data? And then Mark is going to present the tools that are available to you to leverage the data. So here it's really about accessing the data for your own analysis mostly. Um, okay, so first, the license for our data. What can you do with our data? You can do anything you want. So all our data are under the Creative Commons Universal license. So it means that you don't even have to cite us for using our data. So we appreciate if you do, of course. It's recommended that you cite, but it's not even mandatory. And that's because then our data can be integrated in other databases, in other resources. And if there was a more constraining license, there is a problem of license stack, like a resource using a resource using a resource, and then you are supposed to cite everyone. So we cut the problem here by providing a CC0 license, so you can do everything, absolutely everything you want. You can integrate all our data in your own tools, in your own database if you have one. No problem. And yes, please cite us that help us for grant application and things like that, but it's not even mandatory. Okay. So even for commercial application, no problem. Okay, so I will clap just to get you back with us. So Bgee data is so yeah, it's more like a wake-up call after the break. So Bgee data is available upon request, free, available from NCBI. Get stopped now. It's a pretty easy question. Okay, so yeah, fine. Everyone got it right. It's free, available. So yeah, free of charge, free of license, anything you want to do. Okay, so how can you obtain Bgee data? So first you have like download files available from our website. So you have main two types of files. So first you can retrieve the present absent expression calls with the expression score. So the result of the integration of all the data available, okay? So, and we provide different types of files. So first one that is per organ only. So integrating all the developmental stages, all the sexes, all the strains all together to provide you the expression information per organ or tissue or cell type, okay? So per anatomy. And you get a simple file or advanced file. So advanced file contain more columns, basically, with more fine-tuned information, but it's the same rows, the same tissues and cell types are going to be represented, okay? And another flavor is with all condition parameters. So it's really one row is one gene in a tissue at a developmental stage for a specific strain and a specific strain. Okay. So all the condition parameters that we annotate, then they are provided in those files. Okay. So all the data integrated in Bgee, the present absent code and expression score, either per tissue cell type or really tissue stage sex strain. Okay. And then you can retrieve the process data. So if you want to perform your own analysis, retrieving libraries in specific conditions, for instance, so you can use Bgee to retrieve, give me all single cell RNA-Seq performed in the brain in human, for instance. So then you can access what we call the process data. So we provide that for bulk RNA-Seq, full-length single cell data, so SMART-seq, droplet-based single cell data, so 10x, and you can retrieve that per species. So, and for each of these data types, we provide a file listing all the experiments, a file listing all the libraries, and a file providing you with really all the read counts and TPM or CPM values or UMI count. Okay, so you have these different levels like per experiment, per gene. And for single cell data, we provide a H5AD file where you can retrieve this information per cell. And we also provide a TSV file where you can retrieve this information per cell population, because that's how we provide the information in Bgee is by pseudo-bulking the cells per cell population, okay? But you can still retrieve the H5AD file per cell for your own analysis with the annotation of cell type for each cell, okay? But we just don't provide a p-value of expression per cell. We don't provide an expression score per cell. We provide this information per cell population from pseudo-bulking the data, okay? And an example of information you can retrieve. So for the first type of file, so the expression calls, from the integration of all the data available, you're going to retrieve a row is going to be a gene with ID name, an organ, stage, sex, strain, and you're going to get the expression call, the expression p-value, and the expression score, okay? So then you can retrieve this information for all genes in the species and perform your own analysis. For the process data file, so for RNA-Seq data, for instance, you're going to retrieve the experiment ID from SRF, for instance, the library ID, the library type, single N per N reads, and then you're going to have for the gene ID, the gene ID you get the expression value for, and you're going to have the annotation again anyway. So the anatomical entity ID, the stage ID, the sex, the strain, and then you're going to have the quantitative information really coming from the raw data themselves. So you're going to have the read count, the TPM value. We're going to provide the rank information that we use to compute our expression score. And you get a p-value here because it's bulk RNA-seq data. So we can provide a p-value per gene and sample, but it's not the case for droplet-based single-cell data. You won't get a p-value per cell, okay? You will get a p-value per cell population. So it depends. If you retrieve the H5AD file per cell, you won't get a p-value. If you retrieve the TSV file per cell population, you're going to have the p-value. But that's the p-value from the individual samples. It's not the p-value from the integration of all information available in Bgee as in this first expression code file, okay? So process data file is really if you want to perform your own analysis. Give me all libraries in that condition, okay? And then you go your downstream analysis yourself. So to do that, so either you can go on the web interface and download individually the files, or you can use our R package. So we provide a R package available in Bioconductor called BGDB, and it's gonna allow you to do all this data retrieval through R. So through R, you can say, okay, give me all the data available in brain or any substructure of the brain, for instance. And you can retrieve RNA-seq, also Affymetrix data. So you can retrieve the annotations, you can retrieve the process gene expression data, so the read count, the CPM values, you can retrieve from this package. And we provide functions to reformat the data into an expression set object. So expression set is an expression object widely used in many packages to perform expression analysis. So using the BGDBR package, you can retrieve the data, retrieve the process gene expression values, and make that available to other packages for your downstream analysis. So the packages and the documentation are available on Bioconductor. So you can just Google BGDB and you should land on the Bioconductor page, okay? So we don't have time to show you exact use of this package, but you can contact us. There is documentation just so that you know you can integrate everything directly into R. We also provide a Sparkle endpoint, so that's more for like
Sorry, Frédéric, there is a question on the document. Do you use only ensemble gene IDs?
So we prioritize the use of ensemble. So when a genome is available in ensemble, this is the genome annotation we use, and so we provide an ensemble ID. For some species, they are not in ensemble. We use RefSeq for that, and then in that case, we're going to provide a RefSeq ID. So you need to see, so we provide this information of where the genome comes from so that you know the ID, what it refers to. So it's either ensemble or RefSeq. And you retrieve this information on our website or in the package, which ID it is. Okay, so we also provide a Sparkle endpoint. So this is more for data scientists doing semantic web technologies, for instance. So it's more an advanced use, I would say. But the Sparkle endpoint is very convenient to link different databases, different resources together. So using Sparkle queries, you can ask orthology information from a resource with protein annotation from UniProt at the same time as retrieving expression information from Bgee. So you can ask, okay, you can retrieve the organs where a gene is expressed, or all the genes expressed in organ and species, and you can link that to give me the genes expressed in brain and annotated with that protein domain in UniProt, for instance, using a Sparkle endpoint. So you can do aggregated queries. So you can retrieve information on the Bgee website, but we also provide the SIB provide a way to query across all the databases in SIB, basically, or a lot of databases in SIB. So to retrieve the data, you can, so let me check. Yeah. So I'm gonna show you how to use this interface, but we provide an interface when you can query to retrieve three levels of information. So you can query to retrieve experiments, or you can query to retrieve samples, or you can query to retrieve really the gene expression values. Okay. And I'm gonna show you that, and you're gonna be able to play with that interface. Okay. But just keep in mind that there are these three levels of information. And then you can also look at one specific experiment. So if you're interested in GTEC, for instance, or if you're interested in the fly cell atlas, for instance, you can go to a page specific to the experiment where you can download all the files available for that experiment. So that's another way to access to the files available for download it through one specific experiment. And we can also show all the conditions sample in that experiment, okay? Okay, so I'm gonna show you an example, and then I'm gonna let you play with the interface and answer some questions. So if I go back here, so from the Bgee homepage here, you have a link, raw data, okay? And this is the interface with the three level of information I was presenting: experiment level, sample level, and gene expression level, okay? And so I'm gonna do, I don't know, whatever, not the same example as you're gonna do, like, I don't know, like in Drosophila, I want information in the eye. So I submit, so don't forget to press submit here. Okay, and then I'm gonna have like different data types. So I'm gonna have like bulk RNA-Seq data. So I don't have a lot of bulk RNA-Seq data in the Drosophila eye apparently. I have one experiment here, and I have like two single cell experiments here. So I have the fly cell atlas, for instance, and from here I can click to open the page dedicated to that experiment. So the fly cell atlas, you can download the data in H5AD, or you can retrieve the information per cell population, and you can see all the cell types that were annotated in that experiment. And there are a lot currently, because you have like 76 pages for all these cell types, okay? So that's the experiment level. Then if I click, I'm gonna go to the sample level. So apparently I have 8 samples in bulk RNA-Seq with information for the eye. And here I have like libraries. It's all the cell types. So here the 84 rows is cell types per library. Okay, so if you want to see how many libraries you have, you can refine your query here with these filters at the top. So here you can count how many libraries there are, but there are not 84 libraries, okay? Because the 84 rows, it's really cell type per library. Okay, so here for instance, these 4 rows come from the same libraries, but they were studying different cell types. But all those cell types are part of the eye of Drosophila, because that was my query. I requested data in the Drosophila eye. And then I can go also to look at the expression of a gene. So I don't know, a gene in human, I don't know, in a Drosophila, so I know there is brain gene. I don't remember a name of a gene, like, okay, whatever. I take this gene, why not? And I can ask, okay, give me expression level information for that gene in anything that is related to the eye in Drosophila. And then I get expression level from bulk RNA-Seq data. So you can see that we provide the TPM, the read count. You still have the annotation available. And we can go to the single cell data. And single cell data, we're going to provide CPM value. So this gene is not expressed in most of the library. Okay, here you get 2 CPM. And we provide the expression p-value as well. So here you can see that even though we do get 2 CPM, the p-value is very high. So based on the noise from the intergenic regions, we say, oh, you have 2 CPM of expression for that gene in that cell population of ABDB cell type, but actually it's not expressed, it's just noisy expression, okay? So you can see here the advantage of this method of computing true expression signal, because here most people would say, oh, 2 CPM, it's expressed. And if you would go to the library and look at the noise from intergenic regions, you would see that this noise is very high, that probably some intergenic regions did receive 2 CPM of expression value as well, okay? So that's how to play here with these 3 levels of experiment, sample, gene expression level. You have this form here to refine what you want to obtain. So now it's going to be your time to play with this form, okay? So I go to WooClap, and I'm going to ask you to answer a question about the pig brain, okay? A different example of what I did. So first, for the experiment level, so go on the Bgee website. I hide the answer, right? So go on the Bgee website and try to find how many experiments provide data in the pig brain. Okay, so sus scrofa, pig. Is it launch, Mark, the question? I don't know exactly.
Yes, it is.
Yeah. So yeah, you go to the Bgee website, click on raw data here. You are at the experiments level, and you try to identify how many experiments provide data in the pig brain. People are still answering, so I'll wait a little bit. Okay, let's look at the answer. Okay, we get, so those are all the answers that were entered. Okay, so I see a lot of 19, some 18, some very high numbers. So I show you how to do that. So here I'm at the experiment level. I select Suscrofa pig here. I enter brain as a tissue, and here it is selected including substructure, so it means experiment performed really on the brain or any subpart of the brain, okay? And I press submit. And so I see here that I have 18 bulk RNA-seq experiment, and here one single cell experiment with data in the pig brain. So that's why here some people answer 18. They only look at bulk RNA-seq data. You didn't look at the different tab for single cell data. So 19 is the correct answer. 18 bulk RNA-seq, one single cell. This very high number, I suspect they come from clicking that link. Not sure because the computation, so maybe, yeah, so I'm not sure exactly this very high number where they comes from. Maybe if you want to let me know in the chat, or maybe if you want to speak to let me know what did you look at. If you don't dare, it's okay, but you can dare express why you put that number. It's interesting for me to know how you get to that number, okay? So I don't see the chat just in case.
So one person said in the chat, answer to Karen Woklap, many experiments on different platforms. I'm not sure exactly what that means.
Okay.
I have problems with the connection, says the same person.
Anyway, so 19, okay, I hope now it's a bit clearer for you how that works.
Someone put a screenshot. We're putting pig and Uberon. I cannot see the anatomy and the number, but with the screenshot, they get 5 bulk, 41 single cell. So somehow this cannot be pig. I'm not sure what it is.
Species, it's pig. I see. Okay, okay, I see what you did. Okay, thanks. It's because actually you filter through here. So if I go back actually to raw data, what you did is that you filter through here, okay? So here are valid filter indeed, okay? And you did
Ah,
like, right?
someone clicked on guinea pig instead of pig.
Okay. So, but for the
Now it's
screenshot,
okay.
the screenshot is because you apply the filter through here, but here those filters that don't say include substructure. So here these filters are more for refining your queries, okay? So it means that, okay, you perform a query here. So again, like pig brain, oops, pig brain. And then this filter here, they're gonna show you only what is available from your query. So here they're gonna show you only anatomical entities that are part of the brain, okay? And in the first place, they will show you all anatomical entities. And so the problem in your query is that when you look these post filters, they do not include substructure. It's really to say, oh, I would like to see exactly the data in Ammon's Hall. And here I can see only the experiments exactly for Hammond horn. So that's why you didn't get the same numbers, is because when you enter brain here, it was looking only at data mapped to brain and none of their substructure of the brain. Okay? Thanks for sharing. Okay, so next question. How many libraries provide data in the pig brain? Okay? So same annotation to pig brain. But now it's not about the number of experiments, it's about the number of libraries that are available. Don't see people voting. Yeah, it's launched, Mark, book club.
Yeah, it's launched, but it takes a bit of time, I think, for people to find the answer.
Or maybe you did it once with experiments and you don't want to do it again for library. So I wait a few seconds, and if I don't see any answer, I show you. Hope. just it takes time. Fine. Okay, for the sake of time, I'm gonna move on. Okay, sorry for people still trying to answer. Okay, so a lot of 181, some 85, a lot of 18, some 18, 14. Okay, so I show you how to do that. So the most easy solution is like, okay, so, okay, no, I'm gonna do it from scratch again, okay? So libraries, it's like raw data annotation. So I know it's confusing, but we look for a term that accommodates any type of data in situ urbanization, single cell, bulk RNA-Seq. So what we call a sample is different based on these different data types. What we call library is different. So when I asked for number of libraries, it was like bulk single cell libraries here. And so again, I select pig, I enter brain, I submit, up, and I retrieve like 143 bulk libraries. So that's easy. For single cell, it's a bit more complicated because as I told you, the rows, it's per cell type and library. So if you want really to see number of library, you can look at this filter here that really display the actual library IDs if you want to refine your query. So I see only 3 libraries here. So that's a bit of a trick question because it says 38 rows, but it's actually not only the libraries that are displayed here, it's the cell type. So I only have 3 libraries for single cell data and 143. So it's like 146 libraries in the big brain. And so some of you did get the correct answer. So the one person that who find 146, like congrats, it was a trick question. 141, uh, not sure. Sorry, it was like, yeah, 181. Not sure how you get to that number. Probably, yes, summing those two numbers. Yeah, I see. So that was a bit of a trick question. Okay. And then I don't think we have like, oh, wait, I, my Zoom is all messed up because I look at the chat. Okay. I don't think we have enough time so that because I was planning to do it also for genes, but if you look at expression of a specific genes also, you can do the same here. Like so, you select pig, you select brain, but here the difference is that you have to enter a gene. So I don't know. Okay, why not this one? And you press submit, and then you're gonna retrieve the expression information from bulk RNA-seq data and from single cell RNA-seq data as well. So as I show you, the CPM value, the read count, the UMAT count, the expression p-value from that sample. So a lot of p-value really high, showing that even though you get some CPM, it is not expressed here. Expression, the p-value is low enough to say that the gene is expressed. And you see it corresponds to a very high CPM value actually. And that those CPM value here were not corresponding to a true expression. And from bulk RNA-seq data, it's less noisy. So you see like 2 TPM, very confident that it is actually expressed. So you see we have this adaptive threshold, depending on the technology, only thanks to using intergenic regions. Okay. So just to conclude, we also have a JSON API. So it's for the bioinformaticians out there. You can find the documentation at this URL. And you can retrieve the information I mentioned here. Basically, what I show you on the web interface is using the JSON API. So everything I showed here, you can do it programmatically using the JSON API. Okay, so you can access our data through download files, through the web interface or the R package. You can access a Sparkle endpoint, a JSON API, as I just presented the web interface directly. Okay.